Thank you. And initially, I would like to thank the Vebleo. As you told, I'm going to talk today about the effect of gold nanoparticles as potential treatment to cerebral inflammation. This is my mail if you want to contact me after this presentation or so on. Initially, I'm going to make a brief background on nanotechnology. And after that, I'm gonna let you know about the effects of gold nanoparticles in a model of inflammation that we have applied it. That's a cerebral inflammation caused by sepsis. When we talk about nanotechnology, we are talking about a science, engineering, and technology conducted at the nanoscale. What that means? With compounds with size between 1 and 100 nanometers. And what about, what is nanometer? If 1 nanometer was the size of a soccer ball, 1 meter would be the size of the moon. So you can see how tiny is the size of 1 nanometer. And this is a scale with several objects and living beings that we know. And this represents, this clearly show you what 1 nanometer is. Look, this is a tennis ball with 1,000 million nanometers. This is a bacteria with 1,000 nanometer, a virus with 100 nanometer, and 1 molecule of sugar. So we are talking about this scale of size between 1 nanometer and 100 nanometers. And what that is so intriguing about this size, about substance with this size. In this nanoscale, what changes is that the surface area turns really larger. It means that, as you can see here in this figure, if you have 1 square, 1 centimeters side, the total area is 6 centimeters squared. But if you reduce this square in little tiny squares, the surface area is multiplied by 10. It turns 60 centimeters squared. And if you make them even smaller, the total surface area is 6 million centimeters squares. So the surface area is multiplied a lot. Besides, the quantum effects of the electrons that are around this surface is pronounced as well. When we say that the quantum effect is increased, it means that properties like optical properties, magnetic properties, electrical properties, they are all increased as well. This is gold, as everybody knows about gold, with its characteristic yellow color and value. But when we have nano gold, you see that even the color, that's a physical property, is changed. You see nano golds with 20 nanometers in size. If you change a little bit this, 10 nanometers above that, the color is changed as well. 40 nanometers, 60 nanometers, you can see that a physical property that it can be as simple as the color of something, it changes if you change the size of a particle. But not just that. If the physical characteristics of that substance change, other characteristics can change as well. And this is what we observed some years ago. In this paper, we showed that gold nanoparticles, they own anti-inflammatory properties. This was 20 nanometer gold nanoparticles. How we demonstrated that? Using intravital microscopy, that's a system in which we have a anesthetized mice, and we make a surgery to study the tissue, that whatever tissue we want to study. In this case, is brain, okay? But in this paper, we use a tissue called mesentery. And this is the enlarged image that we observed using intravital microscopy. This is a blood vessel in the brain. But as I told you, in this paper, we use mesentery. And this is what we observed. In mice that we treated with water, water is the vehicle of the gold nanoparticles. Gold nanoparticles are dissolved in water. This is why we used water in this group of rats. And we used the other controls, and we used as well one group in which we treated them with gold nanoparticles, citrate-covered gold nanoparticles, 20 nanometer size, or IgG-covered gold nanoparticles. And what we observed, these red arrows, they show where the leukocytes are. You can see that in rats treated with water or water plus IgG, there is a huge increase in leukocytes that migrated through the blood vessel wall. That was not observed in rats previously treated, in rats treated with citrate gold nanoparticles, where the amount of the number of leukocytes that migrated is pretty reduced, as you can see here. So the next question that we asked was, okay, gold nanoparticles, they reduce the number of leukocytes that arrive in an inflammatory site. But gold nanoparticles are useful in any disease. This is when we thought about a disease in which inflammation plays an important role. It's called sepsis associated. In this disease, we induce sepsis in a murine model, and we study brain. And sepsis is a condition in which there is a systemic inflammation caused by infection. And in this model, what we observed is this: we induce infection. There are symptoms of sepsis in all body, including the brain. And the clinical findings that are observed are mainly delirium, seizures, and focal neurological signs. In the clinic, what happens is that along with sepsis, it happens hypotension, hypoxia, and they used to use sedatives. This impairs even more the brain function. And as a result, what happens is that in 70% of people, they die. For those that don't die, what is observed after they leave the health site, after they leave the hospital, is cognitive impairment, depression, and brain atrophy. 1 year to 6 years after they leave the hospital or they leave the health place, it is used to observe these impairments in 33% of the patients. And there are both inflammation involved in the sepsis-associated encephalopathies with release of cytokines, activation of cerebral cells like microglia, and oxidative stress. They all contribute to the sepsis-associated encephalopathy. And treatment of sepsis, it's usually made using intravenous fluid, vasoactive drugs like norepinephrine, antibiotics, but there is no specific treatment for sepsis-associated encephalopathy. So we use it to induce sepsis-associated encephalopathy. We use a model called second ligation and perforation that's represented here, in which we perform 2 punctures in the second. And after this, we treat these animals 2 or 4 hours after sepsis induction with gold nanoparticles. And one group was treated just with saline. And this experiment was performed 6 years after sepsis induction. What we observed was this. In sepsis, in the brain, we observed this in the brain vessels using intravital microscopy. We observed that there is a huge number of leukocyte adhesion in these cerebral blood vessels, as you can see here, compared to the sham group. But in those animals which were treated with citrate gold nanoparticles, you cannot see this inflammation in the brain. Besides this, we observed increased blood-brain barrier failure in sepsis animals, and citrate covered gold nanoparticles prevented that increased blood-brain barrier failure. We observed this injecting Evans blue in the femoral vessel, blood vessel, and we measured the amount of Evans blue that was present in the brain, in the cerebral tissue. Besides, we observed that gold nanoparticles reduced ICAN1 expression in leukocytes, mainly neutrophils, 6 hours of induction of sepsis. What this means? It was observed that ICAN1 is a marker of neutrophil activation. So gold nanoparticles reduced the neutrophil activation in mice that were submitted to this treatment, to the gold nanoparticle treatment. Besides reducing ICAN1 in neutrophils, we observed that gold nanoparticles reduced ICAN1 in blood vessels as well. Here you can see in brown the ICAN1 expression in blood vessels in the brain tissue. But in mice that were treated with citrate gold nanoparticles, the amount of brown is pre-reduced. It means that the ICAN1 expression was reduced on those mice. And as you remember, ICAN1 is one of the molecules that is important for leukocyte extravasation through the blood vessels wall. So reducing ICAN1, you are going to reduce the leukocyte migration. Besides, we observed increased expression in I-kappa beta, phosphorylated in cerebral tissue that was not observed in mice previously treated with citrate gold nanoparticles. I-kappa beta phosphorylation is a marker of NF-kappa beta activation. So NF-kappa beta activation is a transcription factor for inflammatory factors. So if I-kappa beta is phosphorylated, I-kappa beta cannot inhibit NF-kappa beta. This is why gold nanoparticles reduced inflammation in the brain, reducing ICAN1 expression as well. As conclusions, we can say that gold nanoparticles reduce inflammation in brain of mice with sepsis. And besides, gold nanoparticles reduce the ICAN1 expression both in neutrophils and in cerebral blood vessels in mice with sepsis. So gold nanoparticles are a potential treatment for sepsis-associated encephalopathy. This is the people that worked with me to get these results. And these results were possible by the founding of FAPESP here in Sao Paulo from Brazil. Thanks you all.